we are going to talk about the DNA replication. Okay, what we are discussing, what we are going to discuss, DNA replication. Okay, I'm drawing the structure. Just look at here. Okay, suppose that this is the DNA molecules. This is the DNA molecules. Okay, and I told this DNA molecules have the two ends. Suppose that this is the 5' end, this will be the 3' end, and this will be the 3' end, and this will be the 5' end. Clear? So we know very well DNA generally runs anti-parallel to each other in opposite direction. Okay, and this DNA they have the different bases, means they have the nitrogen bases and this bases like that. They present in this way. We'll look at here. They present in this way. This basis present in this way. This is the basis which is found into the DNA molecule. Okay. Now, this ways you can write here A, T, G, C. So then here it will be T, A, C, Z. A, T, Z, C. T, A, C, Z. Okay. So these different bases present in the DNA molecules. Okay. And they are linked with the bonds. So that bonds here you can say they have the A and T double bond and A and T double bond. G and C they have the triple bond. So G and C, I'm going to change the color. Just look at here. G and C they have the triple bond. Okay. So in this way, the bonds. This is the physical structure of DNA molecule. How replication takes place? For the DNA replication, during the DNA replication, there is need of the some enzymes. Okay, some is 5 to 6 enzymes needed for the DNA replication. I am writing the name of enzyme. Just look at that. First one, that is the primase. First enzyme which is known as the primase. Second is the DNA polymerase. Third is the SSBP. That's full name is the single stranded binding protein. Okay, this is the third one. Fourth is the helicase. Okay, helicase, or it is also known as the unwindase. Helicase or unwindase. Fifth is the DNA ligase. Okay, it is also known as the DNA synthetase. Okay, and sixth is the Topoisomerase. Okay, so anyhow you have to learn all these enzymes name. Clear? Primase, DNA polymerase. In DNA polymerase they have the different types. DNA polymerase maybe also have the different types, like here DNA polymerase first, polymerase second, polymerase third. Okay, so DNA polymerase first, DNA polymerase second, and polymerase third. So here DNA polymerase third is the major polymerase enzyme. DNA polymerase third is the major polymerase enzyme. Okay. Clear? Okay. So primase, first one is the primase, second is the DNA polymerase, third is the SSBP, single stranded binding protein, and fourth is the helicase or unwindase, and fifth is the DNA ligase or DNA synthetase, and last one is the Topo Isomerase. Some places they have also given the exonuclease or exonuclease activity. It is also found into the DNA polymerase. Okay, so we are not going to talk separately as a exonuclease activity. Okay, so these are the half dozen of enzyme. Okay, So half dozen of enzymes involved in the during DNA replication phenomenon. Is it fine? And one terminology also used, that is called the replication fork. Replication fork. What means of fork? Fork means seizure. If you have to see the seizure always looking like that. Seizure. If you have used, it will work like that. Okay. So this replication fork, when the DNA molecule, suppose that this is the DNA molecules. Okay. When it is going to open, so that time this DNA molecules, they behaves as like that. They appears like that. Okay. Now look at how it is looking like. Okay. Just look it. It is looking like as a fork. Fork. It is looking like as a fork. Okay. So this portion which I have opened here from here to there. This portion is called as a replication fork. Okay. This portion is known as the replication fork. Fork means seizure. Fork means seizure shape. If you have to see in the seizure, they are looking like that. So this have, if we will compare here, so they appeared like that. So this is called the replication fork. Okay. So this is the important topic, replication fork. Next one, origin of replication, that you can say ORIC. ORIC stands for origin of replication. Origin of replication. Clear? Origin of replication. This is also important because DNA, if you'll check the length of DNA, so DNA is the very long. Okay, that you know very well, the DNA length is 2.2 meter long. So the big problem here, who will define where I will start? Because I gave the DNA and I said where means where you will start DNA replication, because DNA length is too big long. You can't copied whole DNA within one time. You can't copied all the sequence of whole DNA within one time, because we will also discuss the Human Genome Project HGP. That what you have seen in that HGP, it will take too much time, more than 10 to 12 years. They have started, I think, from 1990, I think 1990, and it completed in 2000. Means more than 10 years completed. Okay, for this total sequence of this gene. Okay, so that means, and we are talking about the basis, means the DNA duplication. So where we will start, this has been recommended. Okay, so here, firstly, suppose that this DNA molecule. So primase. First one is the primase. Primase, I told, primase is the enzyme. Primase is the short stretch of RNA molecules. Short stretch of RNA molecules. Clear? What is the primase? Primase is the short stretch of RNA molecules. Are you getting? Primase is primase means enzyme. It is the enzyme which synthesize primase is the enzyme which synthesize short stretch of RNA molecules. Are you getting? Primase is the enzyme which have ability to synthesize the short stretch. Stretch means piece. Stretch means piece of RNA. Clear? Piece of RNA means 6. How many number of that RNA? The number of RNA, how many? That will be 6 to 12. How many nucleotide base pair? It may be 6 to 12. 6 to 12 nucleotide. Okay. So 60 to 12 nucleotide. That is work performed by the primase. So primase is the enzyme which involves in the synthesis of short stretch of RNA molecules. And we know, because DNA is directly unable to initiate the DNA replication. Then they have start, but initiation cannot be occurred by the DNA itself. There is a need of primase enzyme. So primase is very, means you can say utmost important thing. Okay, primase is the utmost important stage of RNA molecule. Okay, let's look at, suppose that this DNA molecules is going to form the replication fork. Okay, it is going to form the replication fork. So here I am drawing the replication fork. Okay, this replication fork. What happened, because this DNA molecules, they are linked with the hydrogen bonds. Here hydrogen bonds, here hydrogen bonds, here hydrogen bonds, here in between they have the hydrogen bonds, different hydrogen bonds is there. One enzyme, and the name of enzyme is the helicase. This is the enzyme, and this name of the enzyme, you can say name of enzyme is the helicase. Helicase, helicase or unwindase. This enzymes involves in the breaking of all hydrogen bonds. It will start to break. It will start to break one by one all hydrogen bonds. Okay, it will start to break one by one all hydrogen bonds. Clear? Okay. So then helicase, when it will come, it will start to open the double stranded DNA, because both strands are intertwined with each other. They are anti-parallel, so intertwined with each other. Clear? Then here, this helicase, it will breaks the all hydrogen bonds which is present between between two anti-parallel strands of DNA molecules. Okay. Ultimately, what happened? Just look at here. They breaks the all hydrogen bonded, and when it will break, so then it will form. Suppose that this is the DNA molecules. They present in this way. When they will start to break one by one, they will start to open in this way. They forms the fork-like structure. They forms the fork-like structure. So this is the fork-like, and this will happen both sides. This will happen also, means you can say it will happen both sides. Just look at here. Wait one second. It will takes place on the both sides. Suppose that this is the DNA. Okay. Their replication fork formed. Sometimes in the DNA replication may be unidirectional or bidirectional. The DNA replication takes place into the both direction. One, it will be start here in this direction. That will be start here in this direction, and another is the in this direction. Because you know this DNA molecules, that if you check the site, so this is the 5' end, and this is the 5' end, so this is the 3' end, and this is the 3' end, so it will be the 5' ends. Okay, no. So similarly also helicase molecules came, this will be opened the all hydrogen bonds. So here hydrogen bonds also found, here hydrogen bonds also present, here also, everywhere hydrogen bonds present. So this hydrogen bonds broken by the helicase. This is the function. Okay, when the hydrogen bonds break, second one, another protein, some protein molecules will come. That protein here, you can say some protein molecules will come which will bind here. Okay, this protein molecules came and bind here. The basic function of protein, I told, this is called the SSBP. SSBP. Anyone, can you tell the name of SSBP full form? Full name of SSBP? Anyone? Do you know? SSBP, I told, this is known as the single stranded. Yes, someone said, very good. Single stranded binding protein. Single stranded binding protein. This is called the SSBP, single stranded bonding. Suppose that this DNA molecules, they forms the fork. So when this helicase molecules, they cut the hydrogen bonds. Okay, hydrogen bond open. So, but DNA molecules, they are the anti-parallel to each other. Clear? They are anti-parallel to each other. If they cut hydrogen bonds, but it will try to rejoin, because DNA is the stable. It will try to again forms the hydrogen bonds. So to prevent the again formation of hydrogen bonds, this function performed by the SSBP. So the function of SSBP, the basic function of SSBP, you can say, the basic function of SSBP, it prevents the reunion of DNA strands. It prevents the reunion of DNA strands. So if hydrogen bonds, sorry, helicase came, it breaks all hydrogen bonds. So then they form the fork-like structure. This fork they can rejoin, reunite. Okay. So that time they will this helicase also call to the SSBP. SSBP, where are you? You will come. You should to come here. Here your work is prominent. So just come. So then SSBP they will get the call from helicase, and now SSBP came. Why we are saying SSBP single stranded? Because they will bind only one strand here. They bind one strand here and another one strand here. Clear? So that's why single stranded binding protein SSBP. Okay, they will prevent the reunion of this DNA strands. Okay, now this is the now replication forms and they have been. Ultimately, what happened? The direction of synthesis will start. Okay, in one side there is the one enzymes will came, and that I told the name of enzyme, it is known as the primase. This primase enzyme came. When the primase enzymes came, it will synthesize the short stretch of RNA molecules. Okay, and the basic function of short stretch molecules, it will provide the 3' OH group. Very important. 3' OH group. The basic function of primase enzyme, it will synthesize the short stretch of RNA molecules, and this short stretch of RNA molecules, it will provide the 3' OH group, free hydroxyl group. When they will get the 3' free hydroxyl group, next upcoming enzyme is DNA polymerase. Here another enzymes will come, and this enzymes is named as the DNA polymerase. DNA polymerase. Okay, DNA polymerase came. When the DNA polymerase came, then it will start to synthesize the DNA molecules. Means now new DNA strand. New DNA. Just wait. Okay. New DNA strand is going to form. Okay. New DNA strands is going to form. This will be the leading strand, continuous strand. Okay, and the direction should be, if the 5' to 3' primes, it will be the 3', that you can say it is the 3'. This will be the 3' to 5'. Okay, this is the one strand. Another, it's opposite similar to also, it's opposite here you can say, but in opposite strand, they have the different primase. Not only one primase present in the different way. One primase, Irandu primase, Moon primase, then another also Null primase. Okay, how many? 4. Clear? One Irandu, Moon, Null. So this primase came. When the primase came, then again they synthesized the short stretch of RNA molecules. Then again synthesize the short stretch of RNA molecules. Clear? Then wait, it synthesized the short stretch of RNA molecules. Okay, this DNA synthesis is going on. Okay, ultimately, and the direction should be clear. The direction here like 3' to 5' in. So this will be the again 3' to 5' in 3' to 5'. Okay. So in this way, synthesis is going on. This DNA replication synthesis is going on. And now this is the overall. So I told what was the work of different enzyme. Another one, I told function helicase. Helicase involves unwinding, means breaking of the hydrogen bond and open the DNA, double stranded of DNA. SSBP prevent reunion. DNA polymerase involves polymerization, means it synthesizes the new DNA strand. Primase synthesize the short stretch of RNA molecules. Primase synthesize the short stretch of molecules. We are missing 2 enzymes, that is DNA ligase and Topo Isomerase. DNA ligase and Topo Isomerase. What is the function? Just look it. We are missing 2 enzymes, that is DNA ligase and Topo Isomerase. Listen. DNA ligase, this is the okay. DNA ligase here will come. In between, they have the NEET. When they synthesize, here DNA polymerase also come. Means just wait. Here DNA polymerase came, they will synthesize two DNA molecules like that. There is the synthesis of DNA molecules. Here polymerase also came, there is the synthesis of DNA molecules. Here polymerase also came, synthesis of DNA. Polymerase also came, synthesis of DNA. Okay, it is fine. But what happened? In between, they have the space, and this space is called the nick. This space is called the nick or gap. This space is called the nick or gap. So in case of the, and this strand, the strands which is continually synthesized, the strands which is continuous synthesize, this is called the leading strand. Leading strand, clear? The strands which is continue, this is called the leading strand. And the strands which have the nick or gap, this is called the discontinuous strand. Discontinuous strand or lagging strand with shade Gauri. Okay, Gauri already shade. Lagging strand, clear? Lagging strand. Okay, lagging strand. And the segment means, here you can say the segment means from here to here. Okay, just look at it. The segment from here to here. This segment is known as the Okazaki fragment. Okay, this fragment is known as the Okazaki fragment. Okay, means Okazaki fragment included primase and polymerase, and this means they have the three things: primase, polymerase, and this thing. So this strand is called the Okazaki fragment. Clear? This strand is known as the Okazaki fragment. Okay. This is known as the Okazaki fragment. Okay. In this way, this DNA replication is going on. Another function, topoisomerase. Here, topoisomerase. What is the function of topoisomerase? Topoisomerase. Another enzyme, topoisomerase. Topoisomerase, when the DNA, suppose that this is the 2 DNA. This is the DNA 2 DNA. When they are going to open, so it's end. This end is going to intertwinkle. Means you can say this. Okay, just wait. This DNA strand, DNA strand is going to intertwined. This DNA strand is going to intertwine. Clear? This DNA strand is going to intertwine. The means they will intertwined with each other, twinkled with each other. Okay. So the basic function of topoisomerase, it will breaks DNA strand, and reveal, clear? Not reveal, that is not reveal, relieve. Sorry, not reveal, relieve. Reveal means express. So you will use the relieve. Relieve tension, relieve tension of dsDNA. Tension of dsDNA. Are you getting what I'm saying? The basic function of the topoisomerase, it will break different this will break the different strands. It will break like that. It will break from here, breaks from here, breaks from here, break from here, and ultimately the DNA, when DNA is going to open, you have to you can do this experiment. If you have the wire, if you start to open middle, if suppose that they have the, you have to wire, if you will start to open in this way, then what happened in corner, the edge of these two wire, what will happen? They will intertwined with each other. Okay, so the intertwined or tension, super coiling, you can say the exact word you can use. That exact word you can say, this is known as the super coiling of DNA. Super coiling of DNA molecules. Super coiling of DNA molecules. Clear? So super coiling of DNA molecules relieved by topoisomerase. Relieved by topoisomerase. Clear? The super coiling in DNA molecules is relieved by the topoisomerase. So in this way, it is the function. Topoisomerase is two different types. One is the topoisomerase first and topoisomerase second. Okay. So how many types? Topoisomerase TI, two different types, TI first, TI first and TI second. Clear? TI first and TI second. TI first, it cuts only the single strand. Clear? TI first, it cuts only the single strand. Okay. Clear? And it means it cuts only the single strand of DNA, single strand of DNA. And the topoisomerase second, it cuts both strands of DNA, both strand of DNA. Clear? It cuts both strand of DNA. And the topoisomerase first is never require the energy, and topoisomerase second, it require the energy. So topoisomerase second in bacteria, topoisomerase second is known as the gyrase. In bacteria, topoisomerase second is known as the gyrase. Okay. In this way, this is the enzyme, and one remaining, that is the DNA ligase. Last enzyme remain, which is known as the DNA ligase. Okay. What is the function of? अरे हमारे बताओ तो तुम चला है काहे कष्ट करता है. So here phone भी नहीं की. Okay, let's look it here. What I'm saying. Okay. Yes. So DNA ligase. We are going to talk about the DNA ligase molecules. DNA ligase, the space. This is space here. Here they have the space. This is called the space. Okay. This is the space or nick. So DNA ligase, here another enzymes will come, and the enzyme's name is the DNA ligase. DNA ligase. Okay. This DNA ligase involves in the ligation. It involves in the ligation, means here it involves in the ligation. You can say it involves in the ligation. Okay. It involves in the ligation. Means the gap which is present between the lagging strands, it will filled by, okay, it is only filled by the this DNA ligase enzyme. So ligase enzymes involves the ligation of Okazaki fragment, sealing of Okazaki fragment. I told DNA ligase also known as the DNA synthetase. DNA ligase also known as the DNA synthetase. Clear? So DNA ligase also known as the DNA synthetase. In this way, DNA duplication takes place. In case of the one strand, that is the 5' to 3' direction, that is the continuous strand, leading strand. Okay. And an opposite way, that is the 3' to 5' in, that is the lagging strand or discontinuous strand. Okay. Lagging strands are okay. So lagging strands or discontinuous strands. Clear? Okay. So this is the overall story about the DNA replication. Did you get it? Did you get it?
Yes, sir.
Okay.
Yes, sir.
Good, Latika. Okay. So in this way, this is the overall story about the DNA replication. In this slide, it is looking too much hectic, means you can't say hectic, that is excessive, but content is superficial, and it is very helpful for you. You have to learn the each enzyme activity and how the DNA replication takes place. In this way, just you have to learn PPT. That's why it is one. At the time of replication, two strands of DNA separates. Now here you can say they are forming the replication fork. Okay. Each strands act as a template, means each strands act as a parental strand. Each strands act as a parental strand. Okay. A new strand is constructed on the each old strand by the complementary base pairing. Means now here you can say there is the formation of new strands takes place. Okay. And these are the complementary. Complementary means suppose that one is the 5' to 3' direction. Another is the 3' to 5' direction. The new strands which is synthesized, it will be also opposite in direction. Means it will be here. Suppose that it is synthesized in this way. This will be 3' to 5 prime, and if it will be synthesized here in this direction, that will be also 3 prime to 5 prime. Okay. In this way, you have to learn the synthesis of DNA molecules always takes place from 5 prime to 3 prime direction. Okay. So in this way, this is the overall story about the DNA replication. Okay. In the next class, I will talk about the Michelson and Stahl experiments, that is semi-conservative mode of DNA replication. This was proved by the Michelson and Stahl. Okay. So thank you for patience and cooperation. I will remain this on para. I will discuss into the next class. Clear? So thank you to all of you.
Thank you.
Okay, Kirti Vasham.